Well guys, in this video tutorial we'll be talking about the fluorescence microscopy, and more importantly we'll be talking about the fluorescence microscopy principle, because I'm not going to talk about the fluorescent microscope and the structures. I'll be just telling you the basic principle behind fluorescence microscopy. We have talked about different other types of microscopy, which are bright field microscopy, which are simple light microscopy. Fluorescence microscopy is also a part of light microscopy, but this is advanced, and the use of fluorescence microscopy in biological systems are even more nowadays, because it helps us to give us proper understanding of the different regions inside the cell, right? Now the idea behind fluorescent microscopy is very basic, and that is, let's say, like all the other type of microscopy we have. For example, say we have a cell. Let's say this is the cell. So the cell is there, and let's say we want to mark this cell with a particular dye. Let's say the dye is here. Let's say this is this black dot which is present in nucleus only. So we put some dye. Now this chemical factor or the dye that we are providing, this dye has a property that is called the fluorescence nature, fluorescence property. Now this dye that we use, which is also called as the fluorescent dye, this simple dye, the idea behind this fluorescence dye is that this dye can receive a particular wavelength light, for example in this case the blue light, and in turn this dye can emit a different color light, in this case the green light, right? So blue light is hitting to this dye, the dye is exciting, and then the dye is releasing the green light, which we can measure, right? And by looking at the image inside the cell, so as a result of this green emission, we can find the green coloration when the dye is present. So by looking at it, we can tell where exactly our specimen is present. So now this fluorescence microscopy is morely used to tag and look at the delicate structure inside the cell, like let's say the actin protein inside the cell, and how actin is arranging, how the actin polymerization is taking place, or microtubule dynamics during cell division. So we can see this specific work or specific functions that are going on inside a living cell. So that's why the fluorescent microscopy is very much handy, because we can use this microscopy for the living cell or living culture imaging. Now let's talk about the principle of this fluorescence microscopy and how this whole process works. To understand this whole process of fluorescence microscopy, you need to understand two fundamental principles. One of this principle is termed as here, let me take a black color, yeah, it is termed as the Jablonski diagram. Jablonski diagram. So this is the Jablonski diagram. According to the theory of Sir Jablonski, is that there are electrons, presence in inside, in different molecules, right? So if you look at here, in this case there are a different state, a different energy state of all those photons. So if you imagine this light that are coming here, because you know the light as a photon, we look at light as a photon in this all type of fluorescence microscopy. So if you imagine this as a photon, this photon have different energy level. So it has a ground state level, which is the basic level, and from this ground state level, if we add some energy from outside, if you put some photon or energy from outside, that photon excites that electron that is present here to the excited state, so the higher state. For example, it is a S0 state, then we have S1, S2, and different energy state for the electron. So that electron will be kicked onto the higher energy state, that's called the excited state, right? So this require energy, and that energy is brought about by the light source or the photon that is hitting here. So the light source, remember, that we provide there. So we provide this light source, it will kick that electron to the excited state. So once the electron is the excited state, now from that excited state, that energy will be released, because this electron will come down to its ground state. So no matter wherever it is, in the whichever energy or excited state it is, it should come down to the ground state. And during this jump from the excited state towards ground state, it releases energy. It releases energy, and this energy in the form of fluorescence light, as you can see here. So if we excite this particular molecule, let's say this molecule is having this beautiful feature. Not all the molecules have this feature. I repeat, not all the molecules have this feature. There are certain molecules, they have this feature. If we excite them with a particular wavelength of light, for example here with this blue colored light, in that case the energy that the electron is kicked to the excited state, and then the electron is coming down to the ground state and it releases some amount of energy out, and that energy that is released in the form of fluorescence. For example, here it is a green coloration, so it's a green fluorescence is released, and we can actually measure this fluorescence, right? Now the idea here is, at the very beginning, when we hit we hit this electron with this light or the energy, at the very beginning, this reaction works very fast. I mean, we can measure it in femtoseconds, that is the 10 to the power minus 15 seconds. This is how fast this is, very much faster. So this process works very fast, and it excites, but during the emission, the energy, some amount of energy is released as heat. That's called the internal conversion. For example, to the same energy level state, these are the same energy level state, for example, some amount of energy is released as heat, they are absorbed as heat, but some of them are released as fluorescence. So the energy that are released as fluorescence, or the emission light, or the emission energy, is less towards compared to the excitation energy which we provide. So whatever energy we provide via photon or light source is excitation energy, that is less, but the wavelength is obviously excitation energy is high, so the wavelength is short here. But now after the emission time, the emission energy is low and the wavelength is high, so it releases higher wavelength fluorescence there, which we can measure, right? So this is the fundamental concept of fluorescence emission. Now if we look at here now about the intensity of this energy as well as the wavelength of the energy, that is for the emission as well as for the excitation, what we can find is that this is a very basic diagram. This is a graph actually. This graph is very much important because in the x-axis we have wavelength, in the y-axis we have intensity, right? So what we can see here is that whatever energy we provide, that is the absorption energy or intensity of the absorption energy, if you look at here, is that intensity is higher because it is in the top, the intensity is higher, but the wavelength is short. So the incident light or the incident energy that we provide there, that is having, so incident light and then emission. So in incident light the intensity is high, but the wavelength obviously is short, definitely because frequency is high, that's why the intensity is high, but the wavelength will be short because they are inversely proportional. For the emission part, the intensity is low and the wavelength is high. Okay, so that's what we can see. The intensity is low, wavelength is high compared to the incident light. So this is the difference between their wavelength. If you look at here, the difference between their wavelength, so you can see here the intensity though sometimes kind of similar, but that there is a huge difference in the wavelength, and the difference in the wavelength of the absorption energy or the absorption light compared with the emission light is called as the Stokes shift. This is according to the scientist who discovered this process, according to his name, Stokes shift. Okay, this is a shift between the wavelength change or the wavelength difference between the absorption light and the emission light. Okay, so you can see these are never the same because the emission light will have higher wavelength. So these are the two basic concepts of any fluorescence microscopy, right? So if we begin with the fluorescence microscopy, this is one basic type which are called as wide field fluorescence microscopy. Except for that, there are other modifications of fluorescence microscopy. There are evolution of fluorescence microscopy because the wide field microscopy will then be modified to what is called confocal microscopy. All of them are, remember, all of these type are fluorescence but slightly modified. So again, there are certain problems in confocal. To encounter that, to solve that, they have other kind. These are called as two photon microscopy, right? So these are all the different types of fluorescence microscopy. We'll be talking about the wide field microscopy in this video. In the next video, we'll be talking about confocal, and then we'll be talking about the two photon microscopy as well. Okay, so now let's look at the principle of how fluorescence microscope actually works. Now, the major understanding of fluorescence microscope is done in the previous section, but if we look at here, this is the light source. The light source can be tungsten lamp or mercury arc lamp, or you can use laser, because lasers are always better option because it's obviously high energy than other light sources which are dimmed. So the idea here is to have the important thing that, for example, let's say this is the sample or the specimen that we have in the slide, which is fixed, which contains the chemical compound which shows us the fluorescence, and those compounds are called fluorescent molecules. For example, fluorophores, for example, let's say quinine is an example of such a chemical compound which emits fluorescence light. Or let's say GFP, or green fluorescent protein, which can be labeled with any other different proteins or different constructions inside the cell to visualize them with the fluorescent microscope. Now the idea here is we need to excite these factors, this GFP or any other fluorescent molecules. We need to excite them with the help of some excitation energy, or with the help of photons that can come from the either light source or laser, with whichever it is. So let's say here from this light source we put the light in there. Now remember, only a particular wavelength of light can excite this chemical compound or the fluorophore. For example, GFP, only a specific wavelength of light can excite this particular type of molecules, right? So for example, here that wavelength which will excite this GFP here is blue colored light, with the wavelength which is the wavelength of blue, right? So that blue light should be passed only. But the light source that we have, it is constructed of seven different types of wavelength of light, different wavelengths. So we need to filter all the other wavelengths out so that only the blue wavelength passes. So for that filtration, we require a type of filter which is called as the excitation filter. As you can see it here, the excitation filter, this one filters other wavelength, only pass that blue wavelength, and it is now passing through the objective. But remember here, once this blue light is passed through this filter, there is a mirror, simple the act of reflection, and this mirror is called as dichroic mirror. Remember, this is a mirror, but this is a normal type of mirror that we look at. This is a specific spatial mirror, right? Why it is spatial? Because normally what we know, in mirror we see reflections of a particular light or anything, right? So if you look at here, this blue light pass and it reflects by dichroic mirror, and it's now passing through the objective, finally to the sample, to the specimen, whatever it is, right? So till now the sample will be excited, just like we've seen in the image in Jablonski diagram. It will be excited to the higher energy state, and then when it comes down to the lower energy state, to the ground state, it emits the fluorescence. Now the fluorescence light here will be of a specific wavelength, and here it is green in color, and the wavelength that we see here is the wavelength of the green light. So this green light will be emitted, and this will be called as the emission spectrum. So this emitted light will again pass the opposite way, that is through this objective, and then it will encounter the dichroic mirror. Now remember, I've told you dichroic mirror is a very specific and special kind of mirror. These mirrors are designed in such a way so that they can reflect a particular wavelength of light, but on the other hand they can allow to pass a particular wavelength of light through this mirror. In this case of this dichroic mirror, it is designed to reflect the blue colored light, the wavelength of blue colored light. On the other hand, it allows the green colored light to pass through the mirror, and that's a very important stage here. because until and unless it allows this green light to pass, again the green light will be reflected and it will not show any image. But in this case, as it allows green light to pass through the dichroic mirror, now it encounters another filter which is called as the emission filter. Now why we require emission filter? To understand this concept, we need to look at the image in multiple dimension, because the thing is, if this is the slide, for example, this is the cell or whatever it is. Now let's say blue light is hitting onto this surface of the sample and green light is coming out. Now as we looking at the images which are three-dimensional, as we are looking at the samples which are three-dimensional, there are different focus plane or focal plane that originates, right? Top, bottom or medium, different focal planes start to originate, and lights that are coming from different focal plane will meet in the different region of this. So this sample can actually scatter light. So according to our idea, if the lights are coming straight like this, it will be very good for this for the camera to detect or the detector to detect. But actually in reality, we'll see the scattering like this. So these are different kinds of scattering of light, and actually most of the lights are most of the energy for this fluorescence light are kind of lost during the scattering or radiation of heat. So to prevent that, actually to collect all these lights together and help them to be arranged together, emission filter is required. An emission filter will allow to pass this particular wavelength of green light only. It will block any other wavelength of light. For example, blue light. If there is any presence of any blue lights or any other light sources from the background ever, that will be filtered by this emission filter. For example, the excitation filter allows to pass the blue light here. Emission filter only allows to pass the green light here, right? The excitation allows the blue light to pass, right? Emission allows green light to pass, and the dichroic mirror reflects blue and passes the green light. So this is the combination. As you can see, this is the most important point of all of this fluorescent microscopy, is this complex of three components: excitation filter, dichroic mirror and emission filter. This is the complex, and this acts as a chamber. If you look at here the chamber in details, you can find here this chamber is the most important part for any fluorescence microscopy. Like this is a unit which is present in wide field fluorescent microscopy, or it is also present in confocal microscopy and also in two photon microscopy. If you look at here, so this is, as you can see, this is the excitation filter, right? Exciter filter or excitation filter filters out all the others except for the blue. So now let's say at the very beginning there are other, so blue is there, red is there, green is there, every light was there at the beginning. But now after this excitation filter, only blue light passes there and blue light is there. So it will be then reflected there, it will be reflected by the dichroic mirror over there, and blue light is on the top here. So sorry, 1 minute. So here it will be on the bottom now to the sample actually. Now from the sample the green light will be emitted. So now the green light will pass through dichroic mirror, as well as if there is any chances of blue light present, that light will also be filtered by the emission filter there, and only green light passes to the detector and detector detects the blue light. Now the interesting fact about this this whole fluorescence microscopy is the quantification, right? So we can actually measure the amount of fluorescence that is coming in. So by measuring the fluorescence, we can actually tell how many proteins are present of the same type that we're dealing with, like, and all these things. We can actually detect all these things there, right? So that is a very interesting fact. And also fluorescence microscopy help us to understand the the contrast of a particular thing, because in either normal microscopy you don't see such a contrast, but in fluorescence microscopy, if you look at here in this complete cell, only the place where we add our dye start to show us the image. Rest of them will be will not show any kind of image. Rest of them will be black. So that is the very idea, good contrast, as well as we can do this beautiful imaging technique with high resolution. Now this is the structure of a microscope. The fluorescence microscope that we see, extremely similar structure like the other light microscope or bright field microscope, but as you can see here, the difference is only between the presence of, there are other certain type of lens, but actually the filter that is present, that is the most important part here. This is the actual construction. The three things, the excitation filter, the dichroic mirror, and the emission filter, the complex of these three things are the most important fact about all of this fluorescence microscopy actually. And let's look at the advantages of fluorescence microscopy. It provides us high contrast imaging because it only provides us the idea of which region of the cell contains the fluorescent dye and which region does not contain it. It has a high specificity value because as we are tagging this chemical components with the cellular compartments or cellular components, it provides us high specificity value. And quantitative imaging, that I've already told you, that we can actually measure what percentage of actins are added with each other and how the microtubule is shrinking, how it's growing, and all this in a quantitative manner. And the most important thing is we can actually image of a live cell or the living cell. That's the very important thing, because actually we can tag those fluorescent dye with the living cell which is growing, which is dividing, and actually we can take various snapshots in the various formation using different types of dye in different time. Then we can actually superimpose them together using monitor and CPU, and finally we can get the idea of what is going on in an animated form actually. And there are multiple versions of this fluorescence microscopy, like, you know, to make this microscopy even better, because fluorescence microscopy, what the problem about this fluorescence microscopy here, disadvantage of this fluorescence microscopy however, is the photo bleaching. The photo bleaching is, as you can see, we start with doing the microscopy of a dendritic cell. As you can see inside, we just tag this actin filaments or the cytoskeleton proteins of the cell with the green fluorescent protein. So rest of them will be black, only the actin filaments are shown. So very specific and also quantitative arrangement. But the problem is, as we are doing this experiment over time, that's with the same chemical agent, with the same GFP protein, over the time it loses the ability for providing the fluorescence light. So as we go on with the same dye over and over through the experiment, what it does actually, that idea is kind of, it is called as photo bleaching. That means the effect of the emission light is kind of, the intensity of emission light is getting very less, and actually camera and detector will not capture it very well, and we cannot see better resolution images in the further cycles over time. So that is a problem with this type of fluorescence microscopy. So for solving this problem, what we use now, we use those kind of chemicals which are having higher resistance against photo bleaching, which can work more cycle than any other general normal chemical compounds. Or we can tag high amount of we can put high amount of fluorescent tag or high amount of fluorescence proteins in different regions inside the cell, so that they continue this process through a long time, right? So these are the troubleshooting for this particular photo bleaching problem. Another thing is that, as you can see, though it is kind of good, we can see a specific region of the cell, but still this is not up to the mark to visualize very tiny structure inside the cell with good resolution. To have more resolution there, because you can see sometimes we can also see some background noises. For example, you can see a lot of things coming out from this area or from this area. You can't actually see much detail about this area. You can just see that the green fluorescence coming in. So the thing is, to remove this particular problem, what we use, we need to create some other modified versions of fluorescence microscopy called as confocal microscopy, which eliminates out all the other scattered lights or scattered emission light that are coming from different planes of the sample, and only selects the particular point which is the point of focus for our understanding, and only allows the light to come from that particular plane, not the any other plane, and which will give us much better understanding. But obviously there are some drawbacks, so we'll be talking about those confocal microscopy in the next discussion. So keep watching the videos and hope you like them. Subscribe, hit the like button, and please put some comments. Thank you.